you guys hear me? Okay. It's 3:30, so I'm going to get started. Just a reminder to sign in case you missed it. Sign-up sheets are where they usually are. We're going to have a couple sets of announcements at the end after Dr. Duca's talk. But yeah, I'd like to introduce our speaker for today. This is Dr. Frank Duca. He's an assistant professor in the School of Animal Comparative and Biomedical Sciences. And he got his Bachelor's of Science and Master's of Science from Penn State University, and his PhD in Physiology and Pathophysiology from Pierre and Marie Curie University in Paris, France. And his lab studies the role of the gut-brain axis in the development of obesity and diabetes, with a particular focus on diet and environmental exposures and how those impact the gut microbiome and gastrointestinal physiology. So thank you, Dr. Duca, for joining us, and we'll get started.
Cool, thank you. Everyone hear me good? Oh, awesome. All right, so thank you for the introduction. Thank you for the opportunity to present to all of you. So I've kept it relatively short on the research side. I don't want to get into a lot of specifics and make it confusing. So I tried to put something together that everyone could follow and understand. And then I also put a lot of how I came to be where I am at the beginning, which I'll kind of go through first. And as I was mentioned, I started my journey at Penn State University. I did my undergraduate in biomedical engineering, and when I went to university, I was really interested in kind of like drug discovery, drug development, and biomedical engineering was a good combination for me because I was really good at math, and I liked that engineering mindset, and there was a lot looking at designing drugs that could be delivered and how the kinetics and all these things happen. So that was a really great major. It was a very small major. It was only 23 graduating in my class. Penn State, for reference, is bigger than U of A. I think it's like 45,000 students, so really small relative to the university. Everyone in that program either goes into industry or goes into med school. I was leaning towards the med school route at that time. I took my MCATs, started applying to medical schools, was doing really well, and then the summer of my junior to senior year, I decided I'd give research a shot, see if I liked it, and I joined this lab that was looking at how the brain can control food intake and obesity and diabetes, and I instantly just really liked the research. I had a really fun time. So I ended up deciding that instead of going into medical school, I wanted to wait, get my master's, try research a little more, see if I really, really liked it, and then kind of decide from there whether I would continue to medical school or try to do maybe the more academia, PhD, postdoc route. So I stayed and got my degree in nutritional sciences. I switched a little bit because biomedical engineering, it was fun, but it was a lot more mechanical focused and less biology, physiology focused than I was hoping for. So nutritional sciences was a much better fit for me because I was always interested in nutrition and exercise and how that affects the body. So I did my master's, and as soon as I started my master's, I just instantly fell in love with research and completely changed my mind and wanted to continue to do a PhD. At that time, I was going to continue on with my mentor who I did my master's with and do my PhD. However, he took a job in France as a director of research at an agricultural institute in France, basically the equivalent of their USDA, the French's National Institute for Agricultural Research. So I moved to France and did my entire PhD in France and speak no French still to this day. That's more of an insult to me because I just could not learn it. But what was really, really neat about this was I got to live in Paris. So this is where I live down here. This is called Cité Universitaire. It's basically just a bunch of huge dorms put together in central Paris that all students who are either studying abroad or going for school there can live in. And they're house specific. So there's like a USA house, a Canada house, a Sweden house, and all the different countries would live in those houses. And it's really, really fun, really cool. You could go to the other houses and everyone would like have like a party at like the Swedish house and then go to the Canada house. And it was a lot of fun. But at that same time, I actually worked here, and this is the National Agricultural Research Institute. It's only about a 20-minute train ride outside of Paris. When people think of France, they only think of Paris and the huge city. But a 20-minute train ride, and you are in as rural country as you can get. the train was basically right around here, so where the stop was, and you'd have to literally walk through farms to get to the building. So that was this huge difference between living in Paris and then working out on the farm. It was really, really cool. I was doing a lot of microbiome research at the time, and that's kind of why I have some microbiome in the research we do today, is from what I learned at that spot. And then a requisite of being a PhD student is you have to decide for some reason you don't ever want to cut your hair and you want to look like crap and grow out a really crappy mustache. So I achieved that, and so did the other PhD student who was with me. So we had a successful PhD in that sense. After that, I wanted to do a postdoc and continue on. So I decided to shift a little more from obesity and go into diabetes research. At the time, there's a lot of overlap in the diseases, but there are significant differences. So I wanted to learn more about diabetes after learning about obesity during my PhD. So I went to Toronto and I worked in the lab with Dr. Tony Lamb at the Toronto General Research Institute, which is right here. We were actually in this big glass building, glass tower here up on like the 12th floor. What was really nice about this is, for those who don't know really what like a postdoc is you have your PhD, so you're kind of respected in the sense that you've got your degree, but you're not quite ready yet to start your own lab and do your own research. So you're in this kind of in-between phase where you have a mentor and you're kind of working on your own projects, but projects that relate to what they're doing in their lab. So it's kind of a mix between your own ideas and their ideas. And Tony, who was my mentor, was an enormous help for me. He taught me so many things on how to run your own lab, how to manage people, how to do your finances, how to come up with good ideas, how to get funding. So I owe a lot of my success to having a really good mentor at that postdoc stage. But also we had a really great team. This is our group above here. There was about 8 or 10 of us, and we were just like best friends. We worked all the time together. And that's a big thing I always tell everyone. When you're doing research, you have to, 1, love the research you're doing, but 2, also really enjoy the people you're with, because science is, I'm sure you guys are well aware, is not a quick thing. It takes time, it takes effort, a lot of hours in the lab, a lot of weekends, long nights, things like that. So if you're with people that you're friends with, it makes it go so much quicker and so much easier. And we would routinely be in the lab till like 9 p.m. on a Friday night, and no one cared, because we were just going to be out with each other on Friday night anyway, so it was kind of the same thing. So we had a really good time. We had a lot of really good publications. What was really, really amazing about being in Toronto was that, for those who aren't aware, that's where insulin was discovered. So type 1 diabetes, before the discovery of insulin, was basically a fatal disease. People could live for a decent amount of time with type 1 diabetes, but eventually they would just succumb to just so much issues with diet and different wasting mechanisms and things like that, where type 1 diabetes was almost literally 100% fatal before the discovery of insulin. And then literally 100 years ago, as of April, insulin was discovered in Toronto by Doctors Banting and Best. And what's really neat is this is the Toronto General Hospital. That's the first hospital that the patients were treated after insulin was collected. And that is the same facade here as this is what it looks like today. So every day when I went into my building, in my lab, to do research for diabetes, I'd have to walk through the same steps that Banting and Best literally walked through to treat the first patients with type 1 diabetes. So it's an extremely humbling moment, and it was just a really good culture there of diabetes research. They've now developed a huge diabetes center, and they have an enormous amount of really good diabetes researchers. So it was a great place to be to kind of study diabetes. The only bad thing here, some of you might know what this is, some of you might not. This is what's called snow, this white stuff here. It sucks, and it's really cold in Toronto, and it's super windy, and it does snow a lot. Being from the East Coast, being from New Jersey, I was kind of tired of the cold weather. I had a really good opportunity, a really good offer to come here, so I started my lab in 2016 at the University of Arizona. And my lab is really interested in gut-to-brain signaling and how that contributes to both obesity and diabetes. And what we're really interested in is, how does the microbiome in your gut interact with what you're eating, your diet, to then alter that gut-to-brain signaling. And I'll kind of go into the gut-to-brain signaling a little more. But first I kind of just want to go into the gut microbiome part, for those who kind of don't know. But the gut microbiome is basically all the microbes in your intestinal tract. So most people just think it's bacteria, but it's not just bacteria. fungus, viruses, any microbe in your gut is considered part of the gut microbiome. We, at my lab, mostly focuses on the bacteria, but overall, estimates say there's about 1 to 10 trillion cells within your gut. So that's anywhere from like 2 to 10 times the amount of cells your body has. So your gut cells outnumber your body cells. And when we're talking about bacteria, there's over 1,000 different species of bacteria within your gut. And what's really neat is those species differ from individual to individual. So it's not like there's only a total of 1,000 species in each individual. There's probably tens and hundreds of thousands of different species and strains that are unique to individuals. It's dominated by two main phyla or divisions, the Firmicutes and the Bacteroides. So people a lot of times will say that there's like this ratio between the amount of Firmicutes to Bacteroides you have, and that could be indicative of either a healthy or an unhealthy gut microbiome, although that's kind of been debated recently. And an important thing for what my lab does is how much bacteria there is in each part of your intestine. So if you think of your GI tract, maybe for those who had physiology, but for those who don't, your GI tract starts, basically if you ingest food, it'll first hit your stomach, and then once it goes from your stomach, it's going to go into your small intestines, so that's your duodenum, into your jejunum, and into your ileum. And this is where most of the nutrients from your food are absorbed. It's going to be in your small intestine, so the duodenum, jejunum, and ileum. And then what's left and what's not absorbed will get into your large intestine, and that's your colon here, and then whatever isn't will be excreted as feces. And just to give you an example of how much bacteria is actually in the human body, 50% of the dry weight of feces is made up of bacteria. So a really gross statistic, but kind of gives you a good example of how much bacteria is really in there. What's interesting though is if you look here, this is the amount of bacteria into each segment. So as you go down the GI tract, there's more and more bacteria in there, where you have a really high amount in the colon, but you don't have as high amount in the small intestine. But as I'll talk to you later, just because there's less of something in there doesn't mean it's not important. And our lab actually focuses on the small intestinal microbiome because that's where a lot of the good physiology is happening. So that's where a lot of that nutrient sensing, nutrient absorption is taking place. And as I kind of mentioned earlier, everyone has a unique microbiome to them. So not one individual's microbiome is not going to be exactly the same as another's. But what is similar is the functionality of that microbiome. So one individual might have a lot of different species than the other, but what those species do is going to be similar from one individual to another. So a lot of important things the microbiome can do are like vitamin synthesis, breakdown of fiber and other diets, activating your immune system. This will be similar from one individual to another, but the actual bacteria that do that are going to be unique to that individual. So where does the microbiome fit in with obesity? So over the last 20 or 30 years, there's been a lot of research coming out that the gut microbiome plays a role in development of obesity and diabetes. So one of the first ways that this was shown was by using germ-free animals. So germ-free animals have no microbes at all. They're completely sterile animals. So they're housed in these bubbles here, and inside those bubbles, everything is completely sterile. There's no microbes, no nothing. So if anyone knows about like a knockout animal, where you can knock out a specific protein in a mouse, and then you can understand what that protein does when you remove it from the mouse, it's a similar concept. You're removing the microbiome from that animal, so you're seeing what happens when they don't have a gut microbiome. And what does happen is they are extremely lean. So mice without a microbiome have a lot less fat than a normal mouse that has a normal gut microbiome, which is really neat. What's even more interesting is if you take them and put them on a high-fat, high-sugar diet, that's similar to how humans will overconsume a high-sugar, high-fat diet and develop obesity. Mice do the exact same thing. They love sugar, they love fat, they'll overeat it and they'll become like these little obese little mice. In germ-free animals, that doesn't happen. They'll overeat and they'll eat the same amount of high-fat diet, but they won't become obese. So the microbiome is playing a role in that development of obesity from high-fat feeding. And what's really cool about these animals is you can actually test different gut microbiomes and see if they are what's driving the development of obesity or staying lean. So what you can do is you can take germ-free animals, and then you can take the microbiome from either a lean individual or an obese individual, and you can inoculate it into those germ-free animals. So these animals are now exactly the same, they're exactly identical, except one animal will have the gut microbiome from, let's say, a lean human, and the other one will have the microbiome from an obese human. And what happens is, in the animals that have the microbiome from the obese human, they develop obesity, whereas those with the lean microbiome won't develop obesity. And what that's telling you is that that microbiome that an obese individual has is contributing to the obesity that they have. Whether it's a cause, that's still unknown, but it is contributing to that obese development. So the other factor there then is there actually is a distinct microbiome between obese and healthy individuals, as well as diabetic and non-diabetic individuals. So this is what's called a principal component analysis. You'll see this a lot with gut microbiome studies or any real big data studies. It's kind of complicated, but there's a really easy way to interpret this data. So each dot here is one subject's entire microbial composition. So it's a way of visualizing thousands of different species at thousands of different levels put as a single dot on this plot. Where it is alone by itself doesn't have much meaning, but what does have meaning is how close or far it is away from these other dots. So this would be one individual's gut microbiome, and this would be another individual's gut microbiome represented as one dot, even though there's going to be thousands and thousands of data points. The closer those dots are to each other, that means the more similar they are to each other. So you can see all these dots here are very close to each other, meaning that their microbiomes are very, very similar. They're not identical, but they're similar. These dots here are very far apart from these dots here, indicating that they're very different. There's a lot of differences. Maybe there's different species in there, or maybe the species are at much different abundances. There's a lot of one, but a little of another. You can see here, the blue are normal weight individuals, so they're all grouping together relatively close, and then you have the obese individuals grouping here on this side. So you can see that they're similar with each other, but they're very different in normal weight and obesity. And you see a similar thing with diabetes, indicating that both these disease states have unique microbiomes that are different from a healthy microbiome. So getting into kind of like obesity and diabetes, I don't know if people have seen these new graphs, but they're pretty amazing. Amazingly scary, I should say, actually. So there's now 700 million people worldwide that are obese and 2 billion people worldwide that are overweight. It's the first time now in the world that there's more overweight people than there are malnourished people, which is a crazy statistic. And then there's this really nice plots here that someone put together about 3 or 4 years ago. And basically this is looking at the percentage of obesity, which would be in blue. So as these boxes get more blue, that's a higher increase in the percentage of that county that's obese. And as they get more red, that's an increase in the percentage of people in that county that are more diabetic. And then as it gets more purple, that's an increase in the combination of obesity and diabetes. So this is starting in 2004, going to 2009, to 2014, to 2019, where you can see even within 15 years, there's almost a 10% increase in obesity and about a 5% increase in diabetes in the country. And this is not stopping. We're at a point now where there will be about 40% of the population will be diabetic in about 10 or 15 years. So extremely high numbers. When we talk about obesity, you can try and really simplify it as a balance in the energy scale where energy intake has to meet energy expenditure. And when you have body weight gain, that means that there's a higher amount of energy intake compared to how much is being expended. So this could be due to a reduction in energy expenditure or an increase in energy intake. And our lab is really interested in the energy intake side. How can you regulate energy intake or control food intake during a meal? However, obesity is not that easy to understand. It's very complex and multifactorial. So the brain is the main center that will regulate energy intake and energy expenditure. And it's always receiving inputs from throughout the body. So it's going to be receiving inputs from the liver, from muscle, from the gut, which we're really interested in, and it'll synthesize all that information and then in real time change energy intake or change energy expenditure. The issue with this is that there's a lot of mechanisms that are contributing to this function that are not within the body. So you have a lot of environment and lifestyle that can affect how the brain can influence intake and energy expenditure. So things like social habits or clock. So when do people eat? Do they eat with friends? Are they eating at buffets? Things like that are going to affect energy intake and energy outtake and how the brain regulates it. Then you have predispositions, so things like early life events that can shape developmental obesity. There's a lot of research right now showing things like mode of delivery of a baby can affect later life development of metabolic disease, or formula feeding versus breastfeeding can actually affect development of metabolic disease. So a lot of early life events, epigenetic effects, can also influence how the brain functions to regulate energy expenditure. But again, our lab is just interested in one aspect, and that's this gut to brain signaling aspect. And we think this is very important because when you just really think in a simple context, the gut is the first site in your body that's going to sense when you're eating a meal, and it's going to realize how much of that meal you're eating and signal that to the brain and the rest of your body to tell your body about the meal you're consuming. So to kind of break that down, you start eating a meal, nutrients get into your small intestine. Before they're absorbed and taken into circulation to be used as energy, they're actually sensed by these special cells called enteroendocrine cells. These cells will sense the nutrients in your gut very early on during a meal. They'll be activated, kind of similar to how a neuron is activated, and they'll release these gut peptides. So mainly GLP-1, CCK, and PYY. These gut peptides will then bind and activate neurons that go from your gut all the way up to your brain. So they'll activate these neurons to tell your brain basically, hey, you have a meal in your gut, you have this much lipids, this much carbohydrates, this much protein. You can start to reduce the amount of food you're eating because you have enough nutrients from that meal. This is a very well-regulated process in healthy humans and healthy animals. So this is an example of how we measure this. So we take mice or rats and we take an intestinal catheter and put it directly into their small intestine, and it comes out the back of their neck. Then we then hook that up to a pump and we'll infuse a nutrient directly into their small intestine. And then once we infuse that nutrient, we'll then give them food and see how much they eat. So in a healthy individual, if we, for instance, give lipids or fat, they'll sense that fat in the intestine as we infuse it into their intestine. So when we give them food, they're not going to eat as much because their body realizes they have those nutrients from that lipid. So it sends a signal to the brain that you don't need to eat as much. Versus if we give them saline, which has no nutrients, they're going to not sense any nutrients. There's going to be no gut-to-brain signal, so they're going to eat more to satisfy the amount of nutrients they need to eat during that meal. What's interesting is on both animals and humans, when they're eating a high-fat diet, this gut-to-brain signaling is very, very impaired. So you don't have this ability of the gut to sense those nutrients. You have a decrease in those peptides that are released. You have a decrease of that signaling to the brain, which is going to lead to overeating or overconsumption during a meal. And chronic overconsumption of meals will lead to weight gain and development of adiposity. So again, we can look at that in our same model. So here again, this is the chow-fed animals. If we give them a saline infusion, it's non-nutritive, so this is how much food they're going to eat. But if we infuse a liquid meal right into their intestine, they sense that liquid meal and they don't eat as much compared to the saline because they can sense those nutrients and signal to the brain to reduce food intake. However, when those animals are on a high-fat diet, you can see there's absolutely no decrease in food intake, indicating that they're not sensing those nutrients in the gut. That's causing overeating, and that overeating will eventually cause weight gain. So if you look at it as a percent suppression, they suppress their food intake when they're healthy on chow, but when they're on a high-fat diet, they don't suppress their food intake. And we're really interested in how is the gut microbiome playing into that, and more specifically, are there ways that we can improve the gut microbiome during obesity and high-fat feeding to restore that gut-to-brain signaling so that you have lower intake during a meal, which will lead to loss of body weight and adiposity. So that brings me to prebiotics. So maybe some of you have heard of probiotics and prebiotics. So probiotics are generally bacteria that are taken, that are beneficial bacteria that might have an effect. But prebiotics are basically the food that healthy bacteria feed off of in your gut. And they're mostly non-digestible carbohydrates, so like dietary fiber. And that dietary fiber can't be broken down by your own body, but the bacteria in your gut can break it down and provide beneficial nutrients that your body can then use. So one of the more common ones that we test is oligofructose. It's pretty commercially available. It's found in things like chicory root, Jerusalem artichokes, mushrooms. But that was one of the first dietary fibers we studied was oligofructose. But we also did a study that was just recently published a few months ago where we looked at all different types of dietary fibers to see which ones were the most beneficial for weight loss. And this is in mice, not in humans. But basically we take mice and we put them on a high-fat diet, so that's going to be the orange lines here. And you can see over time these mice continue to gain weight on the high-fat diet, and that body weight gain is due to an increase in fat or adiposity. We then supplement some of those diets with different dietary fibers. And you can see here there are two fibers that were effective at reducing body weight and reducing adiposity. And that is oligofructose, which is the one we normally study, as well as beta-glucan, which is a new fiber that we're starting to study now. So this is some really nice information that we can then give to the consumer that if you're trying to increase your fiber intake for weight loss, these might be the most beneficial fibers that you should be eating. But that leads to the question as to how are these fibers beneficial? So for us, we want to look again at oligofructose and see, is it working to reduce adiposity and body weight by restoring that gut-to-brain signaling so you don't eat as much during a meal? So what we do is we take rats and we put them on a high-fat diet to make them obese for about 5 weeks, and then we supplement that diet with oligofructose. So they're still on the same high-fat diet. They're still eating the same high-fat, high-sugar diet, except now they have that fiber coming from oligofructose as a supplement in that diet. And when we do that, you can see here that as soon as we start supplementing with the oligofructose, there is a reduction in body weight over time, and that's due to a decrease in adiposity. So they're having a reduction in their fat mass. We can then look at their food intake in real time. We put them in these nice metabolic cages that measures their food intake for 24 hours continuously at every second. And you can see that over time, after the first day, they start to have a decrease in food intake compared to the high-fat diet animals. So they're eating less overall food. We can then look specifically at the meal dynamics when we use these cages and see, is it due to a reduction in the number of meals they eat, or is it due to a reduction in the meal size within that meal? And what we see is that it's actually due to a decrease in meal size. So compared to the high-fat diet animals, the animals with the oligofructose supplement have a significant decrease in the amount of meal size. So that led us again to think about that gut-to-brain signaling, because we know that's how meal size is regulated. But what was important and what was kind of missing in the literature in the field is that all the studies that have looked at prebiotics are always looking at the distal gut intestine, because that's where the majority of bacteria are. So people think that's where the most important part of the microbiome is. However, all that nutrient sensing, all the nutrient absorption, that all takes place in your small intestine. And no one has ever really looked at the small intestinal microbiome. So we hypothesized that that fiber, oligofructose, is altering the small intestinal microbiome to improve that gut-to-brain signaling. So the first thing we wanted to do was to actually look at the small intestinal microbiome and see if it is actually altered. And what we see is a nice example looking in the jejunum. So this is the middle part of the small intestine where a lot of that nutrient signaling as well as nutrient absorption is occurring. And you can see that in the high-fat diet animals here in red, they're grouped together, but they're different from the animals with the oligofructose treatment, indicating that that fiber does shift the gut microbiome. So the next thing we want to see is that shift in the gut microbiome potentially what's driving the improvements in adiposity and body weight? So we can go back to those germ-free mice that I talked about earlier. We have a germ-free facility here at U of A. And what we do is we take the microbiome of the small intestine from either the high-fat diet animal or the high-fat diet animal treated with oligofructose, and we put it into those germ-free mice. So those mice are exactly the same. The only difference in these mice now is one has the microbiome from the oligofructose treatment, and one has the microbiome of just a normal high-fat obese animal. We then can look at body weight and adiposity, and here this is the high-fat animal here, and below it here is the high-fat diet animal that had the oligofructose microbiome. So you can see they do gain less body weight, and that's due to a reduction in adiposity, indicating that the changes we see in the microbiome from that fiber are causing the changes in body weight and adiposity. So now we know the microbiome's changed, we know it has the potential to cause those changes in the phenotype. How is it doing so? So we go back to our model of that gut-to-brain signaling, and we test, can oligofructose restore that gut-to-brain signaling to lower food intake during a meal? To do this again, we take our animals that are put on a high-fat diet for 4 weeks to make them obese, and then we give them a small intestinal catheter. Again, that catheter is going to be directly into their intestine and come out the back of their neck, and then we can hook that up to a pump that can then infuse either a nutrient, in this case was a lipid or fat, or saline, which is a non-nutritive control. So what we do is we fast them overnight, so they're going to be hungry and going to want to eat and consume that meal when we give it to them. But before we give them that meal, we give them either an infusion again of saline as a control or lipids. We then give them their food back, and we measure their food intake for 2 hours. And what we see here is that the baseline is the saline infusion. So in a healthy chow-fed animal, when we infuse that intralipid, that fat, into their gut, they sense it and they decrease their food intake compared to if they had a saline. Basically, they sense it and they're signaling up to the brain to reduce food intake. However, we see no suppression of food intake in those high-fat diet animals, which means they're overeating, and that's causing the development of obesity. But when we switch those animals to a diet with the oligofructose, it completely restores that gut-to-brain signaling to reduce food intake, indicating that that treatment is actually restoring that ability of the gut to sense the lipids. We then want to look to make sure it actually is improving the gut-to-brain part. So first we can measure those peptides that are released from the gut in the circulation in the blood. And you can see in chow-fed animals, when we give that fat into the intestine, there's a significant increase in GLP-1, which is a peptide release from those enteroendocrine cells. It's significantly decreased in those high-fat diet animals, so therefore that's why you're not getting the signal, because the GLP-1 isn't being released. However, when we put them on oligofructose and give them that fat into the gut, they have a significant increase in their GLP-1 secretion. We can then look up at the level of the brain to see if it is increasing the gut-to-brain vagal neuronal signaling. And we look at that through CFOS, which is a marker of neuronal activation. And you can see basically here, there's a lot of activation as represented by these kind of white dots here. These are all different neurons that are being activated in the hindbrain. It's much lower in the high-fat diet animals, but restored when we give the oligofructose. Again, indicating it's restoring that gut-to-brain signaling. Lastly, we want to see if that's actually due to the small intestinal microbiome. So kind of similar to our germ-free transplant, we can do a microbiome swap. So these aren't germ-free animals, these are normal conventional rats that are either on a high-fat diet or a high-fat diet with oligofructose, that the one which can't sense nutrients and the one that can sense nutrients to lower food intake, and then we can swap their microbiomes. And we can have an animal that's on a high-fat diet, has never seen oligofructose or tasted oligofructose, but has the microbiome of an animal that did. And when we transplant that over, it restores the ability of that animal to sense the nutrient and lower food intake. Versus the animal that had oligofructose and normally can sense it, when it gets a high-fat diet microbiome, that high-fat diet microbiome abolishes the ability to sense that nutrient. Overall indicating that what's happening is oligofructose is altering that small intestinal microbiome, and that small intestinal microbiome is then impacting how the host, how the body's intestine can sense those nutrients and signal to the brain. So a really nice host-microbe interaction that we found. We've then kind of gone into looking at some more specifics. So are there certain microbes that are being altered that can potentially be used as a probiotic to bypass having to do like a whole microbiome transplant? So one of the ones we found that was really increased is Bifidobacterium, and specifically it was Bifidobacterium pseudolongum. This was the one that was the most changed throughout all our studies. So we go back to that exact same model, but this time instead of giving oligofructose, that prebiotic, now we're giving a probiotic, which is that Bifidobacterium pseudolongum. So this was the bacteria that the illegal fructose increased. And we do the same fasting refeeding studies, and what we see is in the high-fat diet animals, when we give fat into the gut, they don't sense it, they don't lower their food intake. But in the animals that were treated with Bifidobacterium pseudolongum, it restores the ability of the gut to sense that lipid and lower food intake. Again, now indicating that there's a potential probiotic we can then use and test to treat obese individuals to potentially lower their food intake during the meal. So I'll kind of just stop there. I think it's a good stopping point. And obviously, I have a pretty fun group. I think one of the things I took from being a postdoc and seeing that when you're in a good lab, you can be super productive, is making sure that my lab gets along really well and has a really fun time and enjoys each other's company, and I think we do. We've been pretty successful, and all my students have done really well for themselves and published a lot and gotten their own fellowships, which is really awesome to see. So they're the ones who do all the studies and do all types of different surgeries in all the mice and in the rats, whether it's gut surgeries or brain surgeries. So it's really neat. And we have a lot of good funding sources, which always helps. So with that, I'll take any questions. Yeah.
So I thought in general, horizontal transfer is pretty common to see in terms of like the gut microbiome. So does that kind of indicate that we would be able to see that, like, I guess, like factors of obesity are kind of like contagious per se?
Yeah. Kind of. So contagious, you mean from like one human to the next?
I mean, essentially, yes, because I know that we see like gut microbiome social like differences where they tend to be in groups of people. So does that kind of like apply to this as well?
Yeah, for sure. So they've done some really cool studies looking at like twins who kind of like grow up in different areas, and you can see that there is some genetic influence of what your microbiome is, but there's also a huge social aspect. So families seem to have similar microbiomes within that family versus other families. It's tough to then tease apart, is that due to the family being around each other and kind of just sharing microbes with each other, or is it that they're eating the same meals and it's the food that's ultimately driving it? So the diet is definitely the main driver of the microbiome, but there is a lot with if your family, you're going to share microbiomes. There's even some studies now showing that people can share similar microbiomes with their pets, like their dogs. They seem to see bacteria that show up in both the dog and the human. Not a lot, but there are some.
Okay, so then it's like an extension question. Could this have implications on like the growth of obesity nationwide? Like, do we see that this could like contribute to like an exponential growth in this because there's transfer among people and families and pets and whatnot?
Yeah, for sure. Especially too, because so this is still debated, but the ongoing idea is that in the womb you're actually sterile and you don't have a microbiome. It's not until you're born that you are exposed to microbes for the first time and your intestine develops a gut microbiome. So even you're getting those microbes from your mother. So if your mother's microbiome is dysbiotic, chances are she's going to transfer that microbiome to you. So that's obviously a big thing with an early life event, is the microbiome that you're seeded with. And the type of delivery will cause different seedings of your microbiome. So there's a huge study now looking at cesarean section versus vaginal birth and how that can affect your microbiome, and ways that potentially, if you have to have a cesarean section, can you mimic the microbiome transfer that would be in a natural birth, as well as like breastfeeding versus formula feeding. There's going to be differences there. So yeah, if there's a dysbiotic microbiome that's being passed from generation to generation, it's only going to make it worse.
Thank you so much.
Yeah, no problem. Yeah.
So for the rats, when you took the rats that had the high fat diet and then transferred that microbiome to the sterile ones, did you ever have an experiment where you then gave those sterile rats with the high diet microbiomes with healthy foods and see if that changes the internal microbiome after that?
Yeah, that's a great question. So it actually definitely does. So the diet is one of the main drivers for the microbiome. So a lot of times when we do these germ-free studies, we can only do them for a short period of time, because at first the microbiome will look like the host that you got